So to divide our cellular organisms into prokaryotes and eukaryotes, we can ask the question, does do the cells have a nucleus? And if the answer is no, then we know we have prokaryotes, literally before a nucleus. And then you might recall there's two domains of prokaryotes. So we'd like to ask another question that would let us separate those two domains. So the question we could ask is, does the prokaryote have peptidoglycan in the cell wall? Or do most of the members have peptidoglycan in the cell wall? And if the answer is no, that the prokaryotes cannot make peptidoglycan, that's going to bring us to domain Archaea, the ancient ones, the extremophiles. We back up, and if we say that most of the members in the domain do have peptidoglycan in the cell wall, then that's going to be domain bacteria. And the three members of domain bacteria we'll look at in lab will be Neisseria gonorrhea, the cause of the sexually transmitted infection gonorrhea, Nostoc, which you might recall from our microscopy lab, this beautiful cyanobacterium, and then this is a new one for us. This is the pathogen which causes anthrax, Bacillus Anthracis. And then in the third video, folks, we'll come back and ask, okay, if we have a cellular organism and if they have a nucleus, those will be our eukaryotes, which are all members of domain eukaryote. We'll go over our protists, our kingdom fungi, kingdom plantae, and kingdom animalia. But this part 2 will just be on our prokaryotes. All right, these are our examples that we'll be looking at in lab. So first of all, Neisseria gonorrhea, again, you might recall from the microscope lab when we looked at the urethral smears of a human suffering with gonorrhea. So if we want to classify Neisseria gonorrhea, it's cellular, it's a prokaryote, it belongs to domain bacteria. This is a little bit more information which will make more sense next week when we start doing gram stains. It's classified as a gram-negative bacterium. The individual cells are little ball-shaped cells, and a ball-shaped bacteria cell is called a coccus. Plural is cocci. Often when Neisseria divide in two cells will remain stuck together. And so we refer to that arrangement as diplo. So together we'd say Neisseria gonorrhea often appear as diplococci. This is a stain-like micrograph of possible urethral exudate. So these are these cells with the great big nuclei. These would be human cells, and in these little tiny diplococci, those would be the Neisseria gonorrhea. And here's a light micrograph of a stained urethral smear that looks a lot like the ones that we'll be looking at in lab. And you'll recall the Neisseria gonorrhea triggers a really strong inflammatory response. We're going to attract a lot of PMNs, a lot of neutrophils. The first responders are EMTs. The neutrophils will try to phagocytize, ingest, eat the Neisseria gonorrhea, and destroy them. And here we see again several hard-working neutrophils that have ingested several of the Neisseria gonorrhea. So significance of Neisseria gonorrhea, again, it can cause a sexually transmitted infection, an STI in humans, and the disease is called gonorrhea. It can be successfully treated with antibiotics, and it's really important that we get the patient on antibiotic therapy early so there won't be any permanent damage. And then in addition, folks, this could be a lab exam type question. So in this smear of the urethral exudate, there's the cells of another organism, and that is the human host. So our scientific name is Homo sapiens. And notice, folks, how the genus name is capitalized. The second part of the name, the species or specific epithet, is lowercase. And again, folks, remember with scientific names, we want to make the scientific name different from surrounding text. So when we're using a computer, we can put the scientific name in italics as we have here. So further classification of Homo sapiens, humans, they're cellular, they're eukaryotes, right? Because see all the beautiful nuclei here. They belong to domain Eukarya, and we belong to kingdom Animalia. Okay, and when we talk about human leukocytes, leuko means white, cyte means cells, so white blood cells. They're called white blood cells because they lack hemoglobin. They don't appear red like our red blood cells or erythrocytes would appear. And one specific type of leukocyte that we've talked about here are the neutrophils, the PMNs. This is a new bacterial pathogen, Bacillus Anthracis, and again, folks, this is a scientific name. Notice the genus name has been capitalized, specific epithet or species name is lowercase, right? And we've italicized it. So again, classification, this is all the like lab exam type questions. So Bacillus anthracis, it's cellular, it's a prokaryote, belongs to domain bacteria. And again, folks, next week after we do gram stains, this will make more sense. They are gram positive, and the cell shape, if they look like little sausages or little rods, we call it a single one a bacillus, and more than one, we call bacilli. And furthermore, folks, the little the bacillus right here, and then we see another bacillus right there, right? When Bacillus anthracis divides in two, often the cells remain attached to one another, forming these long chains. And a chain of bacteria cells is referred to as strepto. So if we combine the cell arrangement, strepto, and the cell shape, bacillus, we would call the Bacillus anthracis streptobacilli, forming these long chains. When you're looking at the smears, it might appear as if they're like little hairs, little filaments. But again, if we go down an oil immersion, we can say they're actually chains of cells, these rod-shaped bacilli. And furthermore, folks, just so I won't forget, members of the genus Bacillus are really cool in that they can form these special tough resistant structures called endospores. And these endospores we're going to see can protect the chromosomal DNA of the bacterium for years, decades, maybe even centuries. And in anthrax, the infectious disease caused by Bacillus anthracis, the way we usually get infected is we get exposed to these endospores. We inhale them, we end up with inhalation anthrax, or they get into a cut in our skin and we end up with cutaneous anthrax. Or perhaps we eat tissue from an animal that's died of anthrax, and in ingesting the tissue, we ingest these spores, and then we'll end up with gastrointestinal anthrax. So these endospores, normal boiling won't destroy them. You got to be careful because a lot of antiseptics and disinfectants won't destroy them. Very importantly, folks, alcohol hand sanitizers won't destroy them, but they will be destroyed by normal autoclaving. And you'll recall that's 121 degrees Celsius for 15 to 20 minutes under 1 atmospheric pressure, 15 pounds per square inch. So endospores will be destroyed, but disease-causing prions will not be destroyed by normal autoclaving. The last bacterium is, again, this is kind of a review from the microscope lab. This is nostoc, right? It's cellular, it's a prokaryote, belongs to domain bacteria, and specifically it's a cyanobacterium. So cyanobacteria are bacteria that can make chlorophyll A. And you might recall that chlorophyll A is what provides the nice green color, and chlorophyll A in the presence of light permits the cyanobacteria to carry out oxygenic photosynthesis. They're going to make oxygen and organic molecules like sugars from inorganic carbon dioxide and water. And again, it requires light and chlorophyll A. Now, do remember that nostoc, because they're bacteria, they lack chloroplasts. They have chlorophyll A, but they don't have chloroplasts. And indeed, it was ancient cyanobacteria that evolved into eukaryotic chloroplasts. That's the endosymbiotic theory, you might remember. Now, furthermore, you guys, I hope in some of your slides you'll be able to see two types of nostoc. Now, nostoc is bacteria. They're unicellular, so here's one nostoc cell. Here's another one. They do like to form these nice chains. And you'll notice there's two types of cells here. There's a smaller green cells here. These are the cells that have the chlorophyll A. They're performing oxygenic photosynthesis. So these little cells are pumping out oxygen. But notice here in these chains, sometimes you'll run across a bigger cell that looks different. Different cell, officially that's a hetero site. Hetero means different. And heterocytes have a different function. Their function is not to carry out oxygenic photosynthesis. Instead, the function of the heterocyte is to create an anaerobic environment, no oxygenic photosynthesis, And that anaerobic environment is to protect a really cool enzyme called nitrogenase. And nitrogenase is an enzyme that carries out this cool process called nitrogen fixation. In nitrogen fixation, molecular nitrogen is going to be reduced to ammonia. And the reason this is so important is most organisms, including us humans, we can't break the triple covalent bond that holds the two nitrogen atoms together in molecular nitrogen. Therefore, we can't use all the molecular nitrogen in our atmosphere to build nitrogen-containing organic molecules such as amino acids or proteins or nucleic acids. However, most organisms can use this so-called fixed form of nitrogen, ammonia, or ammonium. We can use that as a source of nitrogen to make amino acids, proteins, and nucleic acids. So we're really beholding to these cool little nitrogen-fixing bacteria that produce this nitrogen that we can actually use to make our own organic molecules. And then furthermore, folks, there's different species of nostoc. Sometimes they make these little globes. They can secrete this little gelatinous mucilaginous sheath here. They can form these cool little globes. And in some cultures, the nostoc are eaten. They're used as a source of food. Okay, let's see here. Down here, let's just take a look at these questions. So, folks, why is the inside of the Hetero Site anaerobic, no oxygen? Right? Well, that's required so the nitrogenase can function. Nitrogenase will be inactivated by molecular oxygen. That's why the inside of the Hetero Site is anaerobic. Right? And again, the nitrogenase, the enzyme that carries out nitrogen fixation, it's going to be inactivated by molecular oxygen. So it can't be active in the same cells that are carrying out oxygenic photosynthesis. Okay, folks, so we'll close this part, and the third part of cast of characters will be the eukaryotes domain eukarya.